Hi loves, it's Miss Huntinko. I hope you're doing well. I want to say thank you to everybody for keeping up with your assignments and contacting me if you need any help. Remember, you can always reach out to me on Remind 101 or through Schoology. Some of you asked me to help you with cell division, and just a reminder that cell division falls under growth on Mr. H. Craig. There are about 4 main things I need you to understand for cell division, and that is that the cell has a life cycle. The purpose of mitosis is so your cells can grow and heal, meaning if you need to make more to, increase body size, or if you have broken cells that need to be fixed. And then the third thing I need you to understand about cell division is binary fission, and that is reproduction in prokaryotes, and that some eukaryotes also use asexual reproduction. And then finally, the last thing I need you to understand about cell division is what allowed us to have multicellular life. All right, so let's review the cell cycle of cells, and these are eukaryotic cells. So we have GAP1, synthesis, and GAP2, and remember that can all be summarized as interphase. At the beginning of interphase, remember if you have 5 chromosomes, once you go through synthesis, you will now have 10. And that's so that when we go through mitosis, that nuclear division, each new daughter cell will get 5 in one and 5 in the other. So you have GAP1, DNA synthesis, and GAP2, which are all summarized by interphase. And then finally, mitosis is specifically the splitting of the chromosomes and the nucleus. And then the very last step that happens at the end of a cell's life cycle is cytokinesis, and that's when they pop apart. Typically, mitosis is only going to happen if that cell is large enough and the DNA is undamaged. Once again, this is a picture of a cell going through mitosis, or that cell division. You've got all the proteins on the outside that help cells talk to each other, and you can see that this is almost complete. Just a reminder about the DNA. We all know that if we've ever moved, you've got to pack everything up. So when the DNA is separating and going into the 2 different daughter nucleuses, it has to be wound up. And remember, this is the original, and then you have the copy, and these are chromatids. If you have 2 chromatids held together at the centromere, then you have sister chromatids. Here's just a recap of mitosis. Remember, that's just nuclear division. Okay, it's the division of those chromosomes in the nucleus. Here you have interphase. You've got a copy of the chromosomes. You go to prophase. That nuclear envelope is disappearing so that those chromosomes can leave. Next, you have metaphase. See, they're all coiled up and lined up into the middle. Anaphase is next. And you see how they're separating away from each other. You have the sister chromatids separating, and those spindle fibers get shorter and shorter. So they're kind of, remember my little prophase, metaphase, anaphase. Okay, so those spindle fibers are getting shorter and pulling those chromosomes to the opposite poles. And then telophase, they're coming back together. At the end, you have 2 new daughter cells. So we start with 1. We're going to end with 2 new daughter cells. So this is our parent cell. And if your parent cell has 10 chromosomes to start with, each daughter cell has to have 10 chromosomes at the end of it. That's so that they can function as normal cells and then go through their own life cycle. Cytokinesis is a little bit different in plant and animal cells. Remember, animal cells, it's called a cleavage furrow. And in plant cells, you have a cell plate that forms. But cytokinesis is actually that splitting of the cells. All right, and you can see here, almost complete. Oops, and just to recap one more time, here you have the cell's life cycle. Just like you have life cycles, cells have life cycles. I need you to know that interphase is a combination of GAP1 synthesis and GAP2, and that synthesis is copying that DNA, and that mitosis is actually splitting of the nucleus, and then cytokinesis splits the cells in half. So here, at the beginning of your life cycle, you have 1 cell, and at the end of it, you'll have 2 new daughter cells that are identical because they need to function again. If you hurt a skin cell, if you get a cut, you need to replace it so that you have, healthy skin cells. If you don't have healthy cells, let's say something has gone wrong in that communication. Ooh, I don't know if you can see me. But uncontrolled cell growth is what we call cancer. So this is when your cells have not divided properly. Typically, your benign tumors, that is just cancerous cells that have not gone anywhere. They stay in a clump that's easy to remove. Malignant tumors are ones that are causing disruption to normal cell functioning and normal tissue functioning. And metastasized means that they have broken away and are infecting another area of the body. So unfortunately, if you know someone who has a cancer that has metastasized, that means it's spread. It maybe started in a lymph node and has spread to the lungs or the brain, or maybe something started in the lungs and now it's spread to other parts of the body. That's what we mean when we say metastasize. Cancer cells don't carry out the functions of the cell anymore. So if you get skin cancer, it doesn't act like a skin cell anymore. It now acts like a cancer cell, and it has unregulated cell growth. Things that cause cancer or cells to lose their normal functioning become cancer cells, we call them carcinogens. Most traditional treatments to treat cancer will destroy both healthy cells and cancer cells, but a lot of the new technology is trying to figure out how to just target those cancer cells. Okay, now on to binary fission. This is very similar to mitosis, but it only occurs in prokaryotes. It looks very similar. You start with one parent cell and you end with 2 daughter cells, and yes, they still need to be identical to the parent, but it only happens in prokaryotes, where mitosis and that cell division is for growth and healing and happens in eukaryotes. Binary fission is for prokaryotes to reproduce through asexual reproduction. I asked you to make a Venn diagram, and so here is a recap of my Venn diagram showing the differences between binary fission and mitosis and what they share. So remember, binary fission, prokaryotes, the whole organism, is a form of asexual reproduction. This can be used by plants, grafting and propagation. And then mitosis is only eukaryotes, and it is that nuclear division. It's in 4 phases: prophase, metaphase, anaphase, telophase. It's used for growth and healing. It is not used for reproduction. Some of the things they have in common, that it is cell division. Remember, prokaryotes are cells, and eukaryotes, that's cells. So we are still dividing cells. We end with identical daughter cells, and also in those processes, the DNA is copied for each. Here's a recap of that asexual reproduction for plants. So fragmentation, if you've ever had a plant and you've kind of cut off a stem, or if you have a succulent and you want to go through this process, you can break off those leaves, let them sit in some damp soil, and they'll start to grow roots and grow new little plants. Grafting is a great way for farmers to use mature root systems and graft a new plant onto an old plant. Why is cell division such a big deal? This is what scientists believe allows us to go from singular life to multicellular organisms. This is just, once we have multicellular organisms and they can go through this cell division, we have the formation of tissues that are all working together on the same function. Once you have tissues that are all working together, those become an organ. So if you have heart cells and then you have cardiac tissue, that cardiac tissue works together to make a heart. And then your heart can't proper function without things like capillaries and arteries and veins, and that would be an organ system. So here's just an example. So we have this level of organization. You go from cells, tissues, to an organ, to a system. So here you have the shoot system that is above ground, and then you have the root system that is below ground. So how do cells become specialized? How do you get cardiac cells from muscle cells to bone cells? It really depends on where that cell was in embryonic development. So wherever those cells are located in the embryo determines what type of cell they're going to be. So whether it's going to be, anybody guess what these are? Those are skin cells. And here you have what your bones look like. And then these are the lining of your intestines. You have these cells that act like little fingers that catch all your food. There's also a lot of bacteria down in there, good bacteria. This brings us to stem cells, and this is just some extra, but you have 3 types of stem cells: totipotent, those can become any; pluripotent, those can grow into anything but a totipotent; and then multipotent. These are all close related. What I really want to talk about with stem cells is that what we are using stem cells now is for treatments, medical treatments. There are a lot of ethical issues depending on where you get the stem cells from. Adult stem cells come from your own body. So that's when you talk about blood marrow transplants. Embryonic stem cells have to unfortunately come from embryos that are grown in a lab and then are destroyed. But one of the good things about embryonic stem cells is they can become any type of cell. All right, any questions, guys? Please reach out to me on Remind 101 or Schoology. I really miss you guys. I can't wait to see you again. I'm so proud of you for keeping up with your assignments. I miss you. I cannot tell you how much that I love y'all, and I love hearing from you. Hope you have a great night, or hope you have a great day. Miss you guys so much.